Thank you. First of all, thank you, organizer, giving me this opportunity to talk about some research work we have done. Also, great to be here, and also great to see you all here. And my name is Zhaohui. I'm from North Dakota State University, and I'm a weed pathologist there, and work a lot on the weed bacterial streak. And because the pathogens are very similar, so I also work a little bit on Barley bacterial streak. Weed bacterial streak arguably in North Dakota could be a longer two disease after scab. It's very pretty economically important in our state. So I just wanted to use this time try to tell you what we've done, try to understanding the host resistance and all the pathogen variants. So my talk will cover, first I will give you a little bit of introduction about the disease and the bacteria, pathogens, and also the previous work on the genetic resistance. And I'll talk a little bit about, kind of give you an overview about the full project I'm doing. We have done or it's been doing, try to understand the genetic resistance and the pathogen virulence. And then I will summarize my work and then our future work is about. And then lastly, we'll give you an acknowledgement in the last. So this disease originally was reported in Barley in 1917, I believe. It's called bacterial blight, and so that disease, and so you can see the symptom here. And so then later we call bacterial leaf streak, and so because of that, the bacteria they form the lesional symptom like a streak, water-soaking streak on the leaf, and a humidity condition. And you will see these kind of bacterial wounds. And so these bacterial wounds can be rain splashed to the up leaf of the plants and then healthy plants, and then disease will be developed to the other places. And when the lesion getting older, you will see this kind of necrotic streak across the cross leaf. Also, those lesions can be coalesces and form large necrotic leaves that get all the leaves get damaged. Highly susceptible varieties, you can see basically the flag leaf will get damaged by the pathogens, and then leading to the yield loss, significant yield loss. And also in wheat, and then also the bacteria also can infect the heads and cause this we call black chef. But probably not much in barley. And the yield impact of this disease has been done, some work done in wheat. And so in the highly susceptible variety, they could suffer about 60% of yield loss. In barley, we still don't have much data on that and how much they can impact the yield. So the pathogen, I'll just give you a little bit about that. The bacteria, so this gram-negative bacteria and rod shape and with a single polar flagellum. And so usually form this kind of yellow mucus colony on the plate if you put on those WBA plates. And so the bacteria has been called a different name. When they was discovered, they called bacterial translucent, and then phytomonas translucent. In the 70s, people classified into the pathogen of captors, Xanthomonas captors. And so then in 90s, kind of reclassify of the Xanthomonas species, then they established Xanthomonas translucent. So right now, I think in Xanthomonas translucent species, they have two groups, we call translucent group, usually contain those bacteria that cause disease on small grains. And so they have three pathogens: XT, pathogen udulusa, XTU, translucent, pathogen translucent, XTT, and pathogen cerealis, XTC. And so they have a different kind of host range. Like XTU can cause disease on wheat, infect the wheat, triticale, barley, rye. And the XTT, the translucent, we usually think just infect the barley, but I can tell you later. I put a question mark there to assume that it's not true. So they can infect other cereal species. And cerealis sounds feel like they have a wide host range. They can infect the cereals, also can infect those grasses, brown grasses. And so then gramineous group, usually those bacteria that cause bacterial wilt on the forage and the pastoral grasses. That's a little bit far away from the translucent group, but in the 90s, since 97.7, you do call, but in the same species, but called different group, translucent group and graminous group. And just last year, I found this is Canadian group. The taxonomists they found that this XTC, the Cerealis, look like is quite different from XTU and XTT, and they think they should establish a new species they call Xanthomonas cerealis, and also the Gramillus, they're also called Xanthomonas gramillus. They're based on the ANI, the cutoff 96%. They think they should establish a new species. I mainly work on the XTU and XTT. XTU cause disease on the wheat, and XTT we think is cause disease on wheat. About genetic resistance in wheat and barley, we know there are not many resistant. I did a lot of disease screening in wheat. We see some, but it's not much. Same way as in the barley. In barley, not many research has been done. So then I just give you list some of the literature that has been showing they're doing some genetic resistance screening and mapping. And so that as early as 1994, I think it's a European group, they find a few lines that is barley lines that have partial resistance. And later they map that resistance in the 3H. And just in the last 2 years, 2023, the Brian Stephenson group, Michi Rittiziger, this PhD student, actually Michi is here today, and they did some screening on many of those breeding lines and cultivars and nanarays, and also including wider integrates. They found very low level of those percentage of lines have resistance, and about only 1.5% have partial resistance. And they also did a kind of GWAS to try to identify genomic region that are associated with resistance. They find 1H, 2H, 6H. So like they have genomic region that are associated with resistance in those lines. So that in the beginning, so we try to do that, and so try to search for host resistance and try to map the resistant QTLs. And so we did that in the Barley mini code. So this work has been done a long time ago, but just don't have, because the data is not a very confident, very balanced. So just recently we just kind of try to do very thorough analysis. I just submitted recently for this paper, but this data experiment was done a long time ago. I just want to give you kind of overview there. The goal for that is just try to identify source resistant and map resistant QTL using the GWAS. And so this is including 198 lines from water body collections. So that's many created by Bob Brookman when he was at NDSU. And he gave me a set of lines, and we planned in the field. And so these lines, those 198, they contain about lines, those cultivars, breeding lines, and nanarays from the 73 countries. And so they captured, based on those marker data, we know they captured the maximum diversity on the water body collections. And so we did a field evaluation in five times in Fargo using natural infection, artificial inoculation. And we also use a 0 to 9 disease rating scales. So this is how they distribute the disease. And so we said, you can see the most of the lines is kind of moderate susceptible to highly susceptible, and so only a few of them, some of them are resistant. Okay. And so then, because of this is called imbalanced data, because some year we don't have those lines, and some year we don't have full replications. And then we tend into transfer into the blob data, and then use that for GWAS. And so this is a list of lines, and so I think it's about 19 lines here, list here. So look like showing consistently resistant across years and across different inoculation methods. I would consider that from 1 to 3 reading is a resistance, and some you can see they have pretty good heading date as the current cultivars, and so it's the bottom several, and you can see they have highly susceptible reactions. And I just give you some kind of visual picture about the reaction in the field. You can see this is a BCN6094. It's very clean when you get into the field, but the other two lines look like highly susceptible. Basically, the leaf is harmed by the disease. Okay. And for association mapping, we download the data, 90K data from the T3 database, and we filtered the marker. We eventually get about 5,568 SNP marker, and we also did a structure analysis, similarly kind of separate into based on the raw type. And so we used Gabbard and did a GWAS, and so test of those 4 different model. And so this is a kind of QQ plot we have there. And so this is Manhattan plot we have. You can see the for each model we find some MTA that are associated with the resistance, and the few of them kind of you can feel those MTA kind of obtained across the different models. So this is just a table, just a list what the marker that's been identified on different chromosomes and showing that they are associated with resistance. The darker ones, that means they are detected by different models. And we can see this 2H1 is kind of major MTA for the resistance. Okay, so that's so that work we've done a long time ago. We just wrapped it up and submitted recently. And the second one, we try to develop kind of marker, PCR-based marker, try to do pathogen detection and quantification. We think that's probably very useful to do a kind of epidemiology study to find out where the pathogen to stay, and also doing quantification for doing kind of seed certification. Maybe we can use those methods. We started from this paper. This was published in 2019. So this Naglais, and they used the comparative genomics to identify region and assume that is very specific to XTU and XTT. And so we find those regions based on the primer and did alignment. Look like this region are very specific to XTU and XTT. And so then we try to develop that PCR-based marker. You can see there. So those regions are absent in those gramineous and in the other denser models. So this is kind of PCR result, and so the T1, T2 is the previous marker was published about the 80s that are using that ITS region, 16S region to try to detect the Xanthomonas translucent. And so you can see they can detect not only XTU, XTT also can detect the other Xanthomonas like those gramineous. And so this is F and R prime look like very specific, just specific to XTU and XCT is they are not amplify anything from those gramineous group. We also try to use that primer marker, try to detect XTT, there's pathogen in the field leaf sample. So we inoculate, we randomly pick those leaf sample that have symptoms, and we took back to the lab and then grind them, and you run PCR. That you can see the use marker can be very efficient to detect those pathogen on those leaf samples. And so these, I think the bright ones, those ones contain a lot of bacterial symptoms, and the rest of one probably contain less symptoms. And also try to detect in barley seeds. We grind those seeds and then try to run PCR on them. Also, you can see they can be used to detect pathogen from the seeds too. Okay. And we also try to detect, try to develop marker that can separate XTT and XTU. XTT, XTU in the culture on the plate, usually you cannot separate them. There's no way you can separate. But you can develop SMP. We develop SMP marker, try to separate XTU, XTT, and also and develop this PCR marker that can very specific to XTT that are not amplifying anything from XTU. And so those markers are very useful too for I think that we give to our Barley pathologist. He did a lot of bacteria isolation from the leaf sample. Look like those markers work pretty well for them. They can verify, oh, that's the XTT or not XTU. We also developed the kind of qPCR, and this is just the figure showing that this primer is very specific, and so only to the region, and also very efficient, and also try to use that, develop those qPCR, try to detect quantify the bacteria in the seeds. So this ST means the standard curve. That we established a standard curve, and then ran the quantification on those samples we got from a field. So this regard to this, I think that's very clean seed we got it from greenhouse, and you can see they have very high CQ value, means they don't have much bacteria in there. But if we got those seeds from field that are infected by the bacteria, you can see they have a lot of, they're very low CQ value, means they have a lot of bacteria in those seeds. Okay. So that's the third project we're trying to do is try to look at the gene expression during the accessibility. So at that time, we haven't found any kind of very resistant barley varieties. So we're thinking maybe we can look at the accessibility, what those genes underlying the accessibility. Maybe we can identify them, and then we can do gene editing, and then making barley become resistant. And so we know in the Zhaohui they produce those are called Tau effectors. So those Tau effectors, they kind of function as when they get into the plant nucleus, they can target the promoter region and turn on those susceptibility gene. And so this is Tau effector has been showing play a very important role in the disease susceptibility in the rice Zhaohui, the Zhaohui or rice cause rice blight. And so we try to see if they have how many Tau in Zhaohui's translucent, and what would they target there to the body susceptibility genes. So first we try to sequence the two Lost Dakota XTT strain. So this the B1FA and the B8G gram GF that come from different state, from different locations. So you can see it compared to there's a type strain, DSM 18974. So that was collected a long time ago, and so was sequenced a while ago. And so you can see our two genome very comparative to that, and we just have a little bit lower lumbar gene, and also a lower probably a little bit smaller than the sequence reference strains. Okay, and we're also using the tools to try to identify to predict the Tau effectors from that Zhaohui. And so this is a RBD sequence of those Tau effectors. So those RBD usually those effectors using those RBD, those amino acids to bind into the promoter region to turn on those susceptibility genes. So based on those RBD sequences, we can predict the target sequence in the host. We try to use that target sequence and try to look at search the body genome to see which gene have that kind of target sequence. Then we also have to do RNA sequence and try to look at those target has been turned on or not. And so that's why we did a kind of transcriptional analysis, and so we use Morris as a subject. And so we use infiltration and have a 3 time point, 1 day, 2 day, 3 day, and also we're using clipping method and day 1, 3, 4, 5, 7, and try to capture those genes. And so for sequencing, we use Illumina HiSeq, and so the very routine those kind of pipeline to do this genome transcription data analysis. And so this is how we got here. This is infiltration. Those DEG was identified. The candle plot, right? This is one plot. So we can see what day 1, day 2, day 3, how many gene is significant differentially expressed, what's a common gene among them. So the right one will be using those clip method we had identified. And so we can see a lot of gene was kind of showing this upregulated downregulate in the filtration method, but not much in the clipping. Eventually, we kind of identified 5 Barley genes, they are highly covalent. Those genes look like they kind of upregulated during those time points, and that they could target by this different toy effector. I think the next step we'll try to do to see if maybe we can do in the CRISPR-Cas9 try to editing those genes to see if they involve in any susceptibility to the bacterial streak. And so in the rice bacterial blight, and so they found their sweet genes very important because they are targeted by those toy effectors induce leading to the disease. We also searched the mox genome, and they find about 22 sweet homologs, and so they're distributed in the different chromosomes. And so we find about 13 sweet genes was expressed for two inoculation methods. And we only find a few of them kind of significantly upregulated for some time point, but not for all time points. They may be involved in the disease susceptibility, but we don't know at this moment. The fourth project we're doing recently, we try to look at the XTT, the host range, and their virulence, how they cause disease using those effectors, and try to determine. We know the XTT, we know cannot cause disease on the wheat, and so we try to determine the wheat and the relatives their reaction to the XTT. They identify those genes in the wheat and relatives, and confirm resistance to XTT. And then we also wanted to identify those type 3 factors that involve in the virulence or virulence. And so usually this you can see if you infiltrate XTU in the wheat barley, and you see water soaking that's showing a susceptible reaction. But if you infiltrate barley strain on wheat and barley, you only see the water soaking on barley, but it's not on the wheat. You usually see this kind of chlorosis, and so that's showing that XTT cannot cause disease on wheat. And so we're thinking is all the wheat and also its relatives also is all resistant to XTT. That's we try to screening the material we have, that including wheat and AMBA wheat and those D genome donor and A genome donor and some other those crosses to see what the reaction is. And so we found for the common wheat, so you usually have this hexploid, right? A, B, B, D, D. They are called homeo chromosome. The durum wheat usually called A, B, B, and the D genome is a DD, and so UR2 is A genome kind of donate for the common wheat, and the monocomb is another A genome species. Ambulata, they have a UU genome, and Cadata, they have a CC genome. So this is a number of accessions list of the sections we test for XTT string from North Dakota. So you can see for common wheat, for durum wheat, and for the dicocum, is we call AMBA wheat, they are all resistant to XTT. They didn't see any susceptibility. So R means they just develop the chlorosis. S means they develop the water soaking, the susceptible reaction. And sometimes you can see some weak water soaking, but with some chlorosis, and also chlorosis with water soaking. You can see I classify that the intermediate. So you can see when we look at the D genome donor Toshii, Agilops Toshii, we filled about 282 lines. And most of them kind of have resistant response. They have kind of one showing very nice water soaking, and so I have 3 that have kind of showing that intermediate reaction. But when you look at the other lookalikes, XT can cause disease on other species. So it seems that it's not XT does not just cause disease on body; they also can cause disease on other wheat kind of relatives. So you can see this very interesting is this AA genome. So look like they have about majority, the A donor UR2, they have majority is kind of susceptible to XTT. But when you look at the monococum, that's also have A genome, look like they have a majority kind of resistant to XTT. But for the caldata, look like they have half and half of that. So this is how they look like. And so this is a torchia. You can see this very strong kind of like crosses resistant reactions. Sorry. And so then the wheat, they have this kind of yellow fluorosis and also have this wall soaking, just showing the kind of this phenotypic that we have. And usually Chinese spring is resistant, right? So we'll try to look at what genes in the wheat is confirmed the resistance. So we're screening those Chinese spring, we call newly tetrasomic lines. So we find is 6A look like contain that resistant genes. And so also when we look at those deletion line, we have this kind of very special deletion line. They can delete a piece of fragment chromosome, they still can survive. We find this 6A, the short arm, if you delete that, they're showing that susceptibility that they indicate those genes probably located in 6A S1, that's breakpoint between 6A1 and 6A5. And so we tried also looking for the effectors to try to recognize, right? Recognized by these resistant plants. So by wheat resistant gene, how they induce the resistance. So we think that, so if we think this type 3 effect is important. So when we lock out those type 3 secretion system, they cannot cause disease and they also cannot cause this chlorosis on wheat anymore. And so this is recently one paper published by, submitted by Jonathan Jackman, Ohio State. They find one effectors that look like they can change the host range. And recently, we also find another one, effector X, will also confirm virulence toward the wheat relatives. So this was first identified in wheat XTU, and then we also find that this is also present in all XTT strains. And so when we lock them out, it looked like they gain some virulence in. So those are different relatives line. That means those lines probably contain that resistant gene to that recognize those effectors. So this is how they look like. So this XTU. If you inoculate effectors, so they become resistant to change from resistant to virulent to the virulent. And so this is other relatives. So we also did a lockouts in the XTT, and then we also, because of the time, and I don't want to spend look at it here anymore. Okay. Just try to summarize, and we find some resistance and doing some RNA sequencing and find some wheat relatives susceptible to XTT. And the next step, I would think, in map to try to clone those resistant in the wheat and try to determine those effectors that play a role in the disease. And I think our research try to provide knowledge and tools for improving body resistance to the BLS. Okay, thank you for your attention.
Thank you, Zhaohui.
Sorry.
I had a question. You were looking for susceptibility factors with your transcription study, is that right?
Yes.
And you used Morex, but wasn't Morex partially resistant? Would you use a more susceptible variety for that?
Yeah, Morex. Yeah, that's a good question. Morex, also it's because we still don't have this very standard evaluation protocol, and we find in greenhouse when you infiltrate XTT in Morex, they're highly susceptible to the XTT strain. So maybe the different strain we use maybe cause the difference.
A good presentation.
Thank you.
So my question is, in your qPCR assay, have you tried to correlate fungal DNA, quantity of the fungal DNA with resistance? If you have done some comparison, is there any correlation between the fungus DNA amplification and resistance?
You mean from other fungal pathogen?
No, in the qPCR assay when you did for BLS, so have you tried to correlate the quantification of fungal DNA, the bacterial DNA quantification with resistance?
Oh, yeah, we found that if we have those variety that showing in the field, they have a lower disease reading when we take the seeds, we find they have less bacterial carryover on the seeds. At the qPCR, yeah, we use that cell, the plate cell we use as to establish that standard curve. We didn't use the DNA. We just used the cell, bacterial cell from the plate culture.
Yeah, so you haven't quantified the bacterial DNA in the host tissue.
Yeah, we try to use that qPCR to quantify bacteria in seeds, also in the leaf samples. Yeah.
Any other questions? No? Okay.